This is your chapter 18 notes. This is part 2. We did part 1 in class. Part 1 talked about gene expression in bacteria, and we went through the trip and the lac operon. In eukaryotic cells, it's actually fairly complex. So let's kind of go step back a little bit and start talking about differentiation. Differentiation is when cells become specialized in early embryonic development. Determination, which is probably a newer word, are the events that lead to observable differentiation in a cell. It makes a cell irreversibly committed to its fate. Each cell contains the entire genome, but only some genes are expressed or actually being transcribed and translated into proteins. We need to activate or repress genes in various cells. Remember, you have the entire genome, but like we talked about in the beginning of class, in this chapter, is we said that just because you make that protein doesn't mean that every single cell needs that protein. For example, insulin isn't something you're going to need in a skin cell, so why would it go through the process of making it if it doesn't need it? That's just wasting energy. We never use some DNA in particular cells. Nerves never make insulin. We use if needed, so a salamander regenerates a lick, so it's only using those genes that are part of that regeneration if they're necessary. We use all the time, turn on when they want to. You can remove cells of a plant to make a new plant. Mitosis clones cells all the time. So there are some that we have and they're there and we turn them on sometimes and we turn them off other times. Come on. This is just showing that differentiation and determination. So the early embryo is going to form, and so there's actually a lot of students ask me, like, how does a cell know what to do? And it's because they're actually sending these signals from one cell to the next cell in a nearby area, and that's telling the cell what genes to turn on and what genes to turn off. And so all the cells in an area will turn on the same genes that will produce the same type of cells and the same type of proteins. So just kind of as going back, we're going to be revisiting some of these things. Just kind of going back, remember you have a gene. This is just a section of DNA. And then it goes through transcription where you make RNA. And then it goes through RNA processing where you add a cap and a tail and you take out the introns and put together the exons. See? Exons are the ones in red that are put together. Then that's going to be transported out of the cytoplasm, and then the mRNA is going to make a polypeptide, which is just a string of amino acids that will then fold into an active protein, and then the mRNA will be degraded. Remember that little animation we did where like the little Pac-Man came and like ate the mRNA? The mRNA will be degraded if it's no longer needed. And then we have the active protein, and then it will go to its cell and it will have some kind of a function, or it can also be degraded if it's no longer needed. So let's start with gene expression with DNA packaging. That's the first kind of level of regulation to gene expression that we have. So a cell's DNA is about 3 meters long. And so here's your DNA. It's wound around these histones. And then those histones are packaged really tightly. A nucleosome is basically a histone with all the DNA wrapped around it. And then this is going to be looped and folded until you have a full chromosome here. So this is the physical drawing, and over here would be a microscopic view. I apologize. Okay. So DNA packaging, histone modification. So the first level, or the first place where we can really control whether a gene is transcribed and translated, has to do with how that DNA is packaged. Acetylation is adding acetyl groups onto the histones. So kind of go back here and you look at these histones. If you add acetyl groups to them, that's called acetylation. Deacetylation is removing the acetyl groups from the histones. Scientists think that acetylation and deacetylation determine how the chromosomes are set up and therefore will influence transcription. Think about if that chromosome is set up in a way that the transcription factors can get in there and easily transcribe and translate, or if they're set up so that it cannot do that. So here's your nucleosome view. These are the histone tails. Your double helix DNA is wrapped around it. And then we add acetyl groups. These are these green things here. Acetylation of histone tails promotes loose chromatin structure and permits transcription. So if that is kind of loose, that means that those transcription factors can easily get to that piece of DNA and actually transcribe it. So acetylation is thought to promote the transcription. The other way that we can do this is adding methyl groups. It's called methylation. So a lot like acetylation, except it's adding methyl groups and not acetyl groups. Scientists have found that the more methylated the DNA, the less active it is, kind of the opposite of acetylation. And they know that because the inactivated X chromosome in females, remember we talked about X inactivation, is heavily methylated. Well, why would it be heavily methylated? It's not being used. Experiments have found that in some species, deficient methylation has led to abnormal embryonic development. It seems that once methylated, these regions continue to be so through additional cell divisions. So the cell keeps a chemical record of this. Once those methyl groups are added, they continue to be there through cell division. This also accounts for genomic imprinting in mammals. We talked about that back in genetics. So here we go. This is your methyl group that you're adding. That's methylation. Okay, so we've got past that and say that it's in favor of being transcribed after the DNA is packaged. Now we can start controlling it through the process of transcription or translation. So transcriptional. Promoters. We think these work a lot like the LAC operon and the TRP operon, where there's something in place that either prevents the RNA polymerase from binding, so that it can't be transcribed, or it's not there and it is transcribed or translated. Activators and enhancers. The binding site on DNA for activators turns on non-adjacent genes when the activator binds to an enhancer, which causes DNA to fold and become adjacent. Kind of this picture here. So it's going to fold and become adjacent, so that's going to help with both of these genes being transcribed. Okay, so now we've got past transcriptional, now post-transcriptional, so right after transcription. So we've already got the gene transcribed. Now, the cap and tail are added to protect the mRNA from being broken down. Remember that little Pac-Man that came and broke down the mRNA? The 5' cap and the poly A tail, they're supposed to help protect that mRNA as it's traveling to get to a ribosome. Gene splicing sequences in the middle of genes are garbage, so we don't want to translate them. Remember, the exons are expressed and the introns are garbage sequences that are taken out. Remember, spliceosome takes those out. Enzymes snip out the introns and put the exons together. So these are other ways of controlling gene expression. You can actually take out pieces of the DNA sequence, those introns, and that's controlling what is and is not transcribed or translated. I think Jonah even asked a question about that in class today. All right, so here's a picture here of those introns being spliced out, and then this is just your coding sequence. This is just another level of being able to control what is and is not transcribed. Okay, so we've got the mRNA now. Now let's look at how do we control translation. How long the mRNA lasts. Okay, the mRNA might be degraded so that it can't be translated anymore. Or it might stick around for a long time so it can be translated. Remember your little animation where you had the little ribosomes, and even when you weren't trying to, the ribosomes were finding the mRNA, and the only way to stop that was to degrade that mRNA. Other proteins bind to inhibit the ribosome. Sometimes ribosomes are blocked so that the mRNA can't be used. Okay, so past the translational. Now we can actually take the polypeptides, or the amino acid sequence, that's the precursor to the protein, into functional units to do jobs on their own. This is an idea that it's not one gene that produces one protein, but one gene produces one polypeptide, and then sometimes they have to come together in order to produce a true functional protein. So that's that quaternary structure. So we have to combine the polypeptides. So if the polypeptides don't combine, that's a control, because at that point the protein is not going to work. It may have been transcribed and translated, but it's not going to be functional because they're not coming together. We can save or break down the proteins, determine how long the protein lasts before it's being broken down. Enzymes can break that up, so proteins can be short-term acting or they can be long-term. Remember, all of this is just having to do with how do we control whether the protein is produced. Non-coding RNAs, genome sequences has revealed that protein-coding DNA accounts for only 1/5 of the human genome. So there's a lot of DNA that we have that doesn't code for anything. A significant amount of genome may be transcribed into non-protein coding RNAs, they're called ncRNAs, and many questions about the functions of these remain, but some may regulate gene expression. They may be there as genes. Remember how I said there's even a gene that has to make the repressor. Well, there are genes that have to control what's going on. And so we're thinking some of those ncRNAs are controlling gene expression. MicroRNAs are complex. They either degrade the target mRNA or block the translation. It's been estimated that expression of at least half of all human genes may be regulated by miRNAs. siRNAs produce similar results to miRNAs. Distinction between how they work is the nature of the precursor molecule for each, okay? But essentially, you can think of it as miRNAs and siRNAs. They're both about either degrading mRNA or blocking translation. Okay, so here's your... So here is your miRNA transcript, and here is the miRNA protein complex. And then if the translation is either going to be blocked or it's going to degrade the mRNA by just existing and being there. A number of steps in gene expression may be regulated during differentiation. That's, remember, early embryo development. Researchers have used muscle cells grown in a lab to help understand this process. So you've got your nucleus, here's your DNA. There's a master regulatory gene called MyoD, and that can actually turn off the transcription factors so that something is not made. The myoblast is a determined, so it's actually a determined gene. It knows what it's going to be. There's an mRNA, and then that can actually produce the MyoD protein. Part of the muscle fiber is fully differentiated cell. And so we can actually turn things on and off using these different mRNAs. This is something that they've kind of come up with the ideas of how this can happen based on their experiments with this particular gene. So you don't have to understand it necessarily, but understand that this is what they've been doing. Like, experiments have shown them how this works. A couple more vocabulary terms you'll need to know. Homeotic genes control other genes that determine developmental sequence. It tells the basic body plan of how everything's going to be laid out. A homeobox is a sequence of homeotic genes that are very similar in many kinds of organisms. This suggests an evolutionary importance. If you look at the early development of most embryos, like early embryo development, they're that early body plan is very, very similar. You can't hardly tell the difference between any of the species. And that's because these are homeobox, or sequences of these genes that are very, very similar. We say that they are highly conserved. Okay, and then the last part we're going to talk about is how cancer relates to gene expression. Genes that normally, and I think one of your readings has to do with that. Genes that normally regulate cell growth and division during the cell cycle include genes for growth factors, their receptors, and the intercellular molecules of signaling pathways. Mutations that alter any of these genes in somatic cells can lead to cancer, because it's altering genes in growth factors, which regulate cell division, the receptors that regulate that, or other molecules that are part of that signaling pathway. Think about cell communication. If the cells are communicating with each other and they're sending bad signals, then they're all going to be bad, and that can lead to cancer. Oncogenes, we've already talked about these a little bit. I'm going to kind of bring them back up. Oncogenes are cancer-causing genes. Proto-oncogenes are normal versions of the gene that code for proteins that stimulate cell growth and division. An oncogene arises from a genetic change that leads to an increase either in the amount of proto-oncogenes production or in the intrinsic activity of each protein. So proto-oncogenes are normal genes. They can be converted to oncogenes by mutations. The genetic changes that convert a proto-oncogene into oncogenes fall into three main categories. Movement of DNA within the genome, amplification of the proto-oncogene, so making more of it, or point mutations in a control element or a proto-oncogene itself. So that's just a mutation. Like, remember those point mutations: substitution, insertion, deletion. Cancer cells are frequently found to contain chromosomes that have broken or rejoined incorrectly. So the gene sequence is kind of out of place. So here's a proto-oncogene. Translocation or transposition, so the gene is moved to a new area on the chromosome. So you've got normal growth stimulating protein is in excess. Gene amplification, so now we're making more just because we're making more of it, we're amplifying it. And a point mutation means that we now have a mutation that could lead to an oncogene. And these are just, if you remember, some of the chromosomal mutations that can happen. And these mutations can be bad because they rearrange the order of genes as they are transcribed and translated. Tumor suppressor genes, genes whose normal products inhibit cell division, normal products inhibit. These are the genes you want to have. They suppress tumors. Any mutation that decreases the normal activity of this gene or this tumor suppressor protein may contribute to the onset of cancer. It stimulates growth through the absence of suppression. Wrap your head around that. So it stimulates growth because it's not being suppressed. Some tumor suppressor proteins repair damaged DNA, which prevents a cell from accumulating cancer-causing mutations. There are a couple of genes you should know about. The RAS proto-oncogene results in a protein that stimulates the cell cycle but needs to be triggered by a growth factor. Certain mutations trigger it even in the absence of the growth factor, leading to an increased cell division. So it's a proto-oncogene that can turn into an oncogene. P53 is a really highly known tumor suppressor gene. The protein results in a specific transcription factor that promotes the synthesis of the cell cycle, inhibiting protein. It activates suicide genes, remember apoptosis, in response to mutations, bringing about apoptosis. So mutation happens, and this gene is actually going to trigger the cell to die so that it doesn't pass that mutation on. Mutations in RAS occur in about 30% of human cancers, and mutations in P53 occur in more than 50% of human cancers. So they're really seeing a link between these two genes and cancer. And this just kind of shows a little bit of how that might happen with the RAS protein and stuff. And then inhibiting the cell cycle pathway. So you can kind of look at that to try to understand what I was talking about before. Cancer can result from an accumulation of mutations, and mutations occur over time. So the longer we live, the more likely we are to get cancer. Mutations contribute to cancer, but we still don't know exactly how a particular cell, that's important here, we still don't know exactly how a particular cell becomes a cancer cell. We kind of understand the progression now, but we don't really understand what triggers that first cell. Development of a malignant tumor is paralleled by a gradual accumulation of mutations that convert proto-oncogenes to oncogenes and knock out the tumor suppressor genes. So basically what we're getting here is a tumor goes from mutations that convert proto-oncogenes to oncogenes, and we're getting rid of the tumor suppressor genes. And usually an accumulation of all of that is what ultimately leads to cancer. And this just shows normal epithelial colon. This is a colon down here. Normal cell growth. You may have heard of colon cancer. And then we have a loss of the tumor suppressor gene or something else. And now we have a little benign growth, a polyp. Those are often removed during colonoscopies. Now we have a second thing that happens to the individual. Now all of a sudden they have an activation of a RAS oncogene. So now we also lost the tumor suppressor gene DCC. Now our growth, now our growth has become larger, okay? And now we've also lost the P53 tumor suppressor gene, and we have additional mutations. Now all of a sudden we've accumulated all of this damage, 1, 2, 3, 4, and 5, and now we have a malignant tumor, and that's going to be a carcinoma. That means it's actually going to get into the bloodstream and it's going to pass that on. This is the point at which a colonoscopy is really helpful, because in the colonoscopy they can actually remove those polyps and they won't develop into that malignant tumor. That's why people over the age of 40 should have colon cancer screenings pretty frequently, because they can catch those polyps, get rid of them, and then they typically are fine from there. But if they don't get rid of them, they have the opportunity to then become malignant. An individual that inherits an oncogene or a mutant allele of a tumor suppressor gene is one step closer to accumulating the necessary mutations for cancer to develop. Just because you inherit a bad tumor suppressor gene or an oncogene doesn't mean you will get cancer. It just means you're one step closer than the rest of the population that doesn't have this. BRCA1 and BRCA2 are genes associated with increased susceptibility to breast cancer. Mutations in at least one of these two genes are associated by at least half of inherited breast cancers. Both considered tumor suppressor genes because their wild type alleles and protect against breast cancer, and their mutant alleles are recessive. Both protein products function in the cell's DNA repair pathway. Let's not forget that DNA damage can also be caused by external factors. You might not just inherit a gene that is damaged. Smoking damages your DNA, UV light, chemicals, radiation, that can all damage those genes from the environmental things that can damage those genes. And so it makes sense to try to minimize those exposures to the environment. You can't do anything about your genetics, but you can minimize your environmental exposure. Some known viruses have been associated with cancers. Epstein-Barr viruses that causes mononucleosis is linked to Burkitt's lymphoma. The HPV virus is linked to cervical cancer. There is actually now a vaccine for HPV. You may have heard of it. You may have had it. That didn't exist when I was your age. HTLV1 virus is linked to a type of adult leukemia. Viral integration may donate an oncogene to a cell, disrupt the tumor suppressor gene, or convert a proto-oncogene to an oncogene. So how does the virus cause that? Because it can disrupt these genes. And that is the end of that. And I know it was a little bit longer. No, discard.